The dream of generating tissues and organs for transplantation into humans.
It began with simple breeding, choosing the strongest, the fastest, the most beautiful. But over time, humanity's desire to control life grew beyond the fields and farms, reaching into laboratories where genes could be edited and species combined. But in the pursuit of perfection, we've created creatures that nature itself would never allow. Cows that look like bodybuilders, fish that glow in the dark, and goats that spin silk in their milk. And these are your spider goats. These are the spider goats. Humanity's tampering with animal genetics has created some truly unsettling results. These are the 20 most terrifying animals humans have bred into existence. 20. Belgian blue cow. The Belgian blue cow looks like it spends every waking moment at the gym with bulging muscles that seem anatomically impossible. Its body is a grotesque testament to human obsession, an animal sculpted into something almost monstrous. The breed originates from Belgium, where farmers selectively bred cattle carrying a genetic mutation in the myostatin gene. This deletion causes what scientists call double muzzling, where muscle fibers multiply instead of simply growing larger. The results are dramatic. Belgian blues produce about 20 more muscle mass than normal cattle with extremely lean meat highly priced by consumers. But this impressive physique comes at a terrible cost to the animals themselves. The mutation creates severe health complications that plague these cattle throughout their lives. Their massive muscle development leads to impaired mobility, joint problems, and frequent injuries. The most disturbing issue involves reproduction. Female Belgian blues routinely experience dangerous sieving difficulties because their narrow birth canals can't accommodate the large, heavily muscled calves. Caesarean sections have become standard practice, with cows sometimes enduring 5 or 6 surgical deliveries during their lifetime. Each birth is a brutal reminder of how far we've pushed nature's limits. Life brought into the world through pain, precision, and quiet horror. 19. Glowfish. They appear almost otherworldly. Zebrafish swimming in aquarium tanks glowing in brilliant reds, greens, and oranges under black light. Glowfish were created in the late 1990s by scientists at the National University of Singapore, who inserted fluorescent protein genes from jellyfish and sea anemones into zebrafish embryos. The original purpose was detecting water pollution, but these genetically modified fish quickly became the first genetically modified animal available as pets. The FDA approved them in 2003, determining they pose no threat to the food supply or environment. That assessment has proven problematic. In Brazil, where glowfish are banned, escaped fish from illegal breeding operations have established wild populations in at least 5 different waterways. Researchers discovered these fluorescent fish breeding successfully in creeks within the Atlantic forest, one of Earth's most biodiverse regions. The ecological implications are alarming. These glowing fish invade ecosystems like living neon intruders, outco-competing native species, drawing predators with their unnatural glow and throwing entire food chains into chaos. It's as if nature itself can't look away from the mistake we've unleashed. 18. Spider goats. Imagine a goat that produces spider silk in its milk. At the University of Wyoming, researchers engineered goats whose very DNA was fused with spider silk genes, creating living hybrids that blur the line between creature and creation. The process involves inserting the dragline silk gene from golden orb weaver spiders directly into goat embryos, targeting the mammary glands. When these genetically modified goats mature and lactate, their milk contains spider silk proteins that can be extracted and processed into incredibly strong fibers. Spider silk possesses tensile strength 5 times greater than steel and 3 times stronger than Kevlar, making it valuable for medical sutures, artificial tendons, bulletproof vests, and military applications. The biotechnology company Nexia originally pioneered this work in 2002, dubbing the silk protein BioL. When Nexia went bankrupt in 2009, Dr. Randy Lewis at Utah State University continued the research. His facility now maintains over 20 spider goats with approximately 3 out of every 7 offspring carrying the silk gene. The spider goats appear healthy and behave normally, showing no obvious health problems after 15 years of breeding. But the idea of fusing creatures from entirely different worlds raises chilling questions about how far humans are willing to go in defying nature's boundaries. 17. Featherless chicken. Israeli geneticist Avagdor Kahan spent 50 years breeding what might be the most unsettling bird ever created. The featherless chicken stands completely naked. Its pink wrinkled skin fully exposed without a single feather for protection. Kahana crossbreed broiler chickens with naturally featherless neck breeds, gradually amplifying the trait through 6 generations of selective breeding. His goal was creating chickens that could thrive in hot climates without expensive cooling systems while eliminating the time and cost of plucking during processing. What emerged was a creature that disturbs even experienced farmers. Without feathers, these birds suffer from sunburn, temperature regulation problems, and extreme vulnerability to parasites and skin infections. Their exposed skin is highly sensitive and prone to injuries. Mating becomes dangerous because roosters lack feathers to grip during breeding, leading to scratches and wounds on hens. Animal welfare organizations strongly oppose the breed. Critics argue it exemplifies the worst of industrial agriculture, prioritizing processing efficiency over animal well-being. The featherless chickens require vigilant veterinary care and cannot engage in natural behaviors like pining and dust bathing. A chicken like this will put on more than 100 g of weight a day, which are essential for normal chicken psychology. Russian veterinary experts warned that these birds live in a state of constant discomfort. Their lives cut short by the very design meant to make them better. 16. Africanized honeybee. In 1956, Brazilian geneticist Warwick Kerr attempted to create the perfect honey-producing bee by crossbreeding African and European honeybees. He aimed to combine African bees' heat tolerance with European bees' gentle temperament. The experiment went catastrophically wrong in 1957 when a visiting beekeeper accidentally released 26 African queen bees from containment. Those escaped queens bred with local European populations, creating hybrids that retained the worst traits of their African ancestors. The resulting Africanized honeybees, dubbed killer bees, spread across South America at 200 to 300 m per year. They reached Texas by 1990 and now inhabit the southern United States. Africanized bees are significantly more defensive than European honeybees, reacting to disturbances 10 times faster and pursuing perceived threats up to a quarter mile. They attack in overwhelming numbers. Victims receive 10 times more stings than from European bees. These killer bees have caused approximately 1,000 human deaths since their creation. 15. Aquadvantage salmon. In 2015, the FDA approved the unthinkable, the first genetically engineered animal for human consumption. AquaBounty's Aqua Advantage salmon carries genes from a chinook and an ocean pout, forcing it to grow endlessly. One small company is producing a genetically engineered salmon it says could help solve some of these problems. It's a fish built for profit, not nature, a living mutation born from our hunger to control life itself. The modification allows these fish to produce growth hormone year-round instead of only during spring and summer. Aquadvantage salmon reach market size in 16 to 18 months, half the time required by normal Atlantic salmon. Atlantic salmon modified to grow faster. They grow twice as fast while consuming 25 less feed, promising environmental benefits and economic advantages. But critics raise serious concerns about long-term implications. Environmental groups fear that if these GM fish escape into oceans, they could outcompete wild salmon populations. Although engineered to be sterile females, no sterilization method is 100% effective. The FDA requires Aquadvantage salmon raised only in land-based contained facilities with multiple barriers preventing escape. Because the fish can be labeled simply as Atlantic salmon, many consumers unknowingly eat it, representing both promise and peril of genetic engineering. 14. Bully Whippet. Whippets are elegant racing dogs known for their slim, athletic build. But some whippets are born with a shocking mutation that transforms them into heavily muscled giants. These bully whippets carry 2 copies of a deletion in the myostatin gene. The genetic defect causes extreme muscle hypertrophy throughout the body. Bully whippets can barely walk properly under their own muscle mass. Their bodies grotesquely enlarge beyond natural proportions. Their necks, chests, and thighs bulge with excessive muscle, serving no functional purpose. These heterozygous dogs are faster racers than normal whippets, creating competitive advantages that may have driven the mutation's persistence in racing bloodlines. But bully whippets suffer greatly. They experience muscle cramps in their shoulders and thighs, often develop jaw overbites, and face mobility limitations. Even with proper care, many of these dogs are sentenced to death. Stripped of value by breeders for failing to race, their lives are ended mercilessly, discarded as if they were nothing more than broken objects. 13. English Bulldog. The English Bulldog has become one of the most popular breeds in America. Instantly recognizable with its push-it-in face, wrinkled skin, and squat body, this beloved breed is also one of the sickliest animals humans have ever created. Centuries of selective breeding transformed original athletic bull-baiting dogs into drastically deformed companions. Modern bulldogs stem from just 68 founding animals, creating an extremely limited gene pool. Breeders selected for increasingly exaggerated features that owners find adorable but veterinarians call catastrophic. The shortened skull causes brachycephalic syndrome, meaning bulldogs struggle to breathe normally. Their compressed airways force them to work harder for every breath. Many cannot run without gasping. Bulldogs suffer from skin fold infections, eye problems, hip dysplasia affecting 72% of the breed, and heart defects. Most cannot mate naturally and require artificial insemination. Over 90 need cesarean sections because puppies' heads are too large. Less than 10 live past 8 years old. Genetic analysis reveals bulldogs have lost so much diversity that restoring their health may be beyond reach. A living nightmare of a species trapped by its own engineered flaws. 12. Liger. When a male lion mates with a female tiger, the result is a liger. The colossal unnatural hybrid that towers as the largest cat on Earth. A living testament to the terrifying extremes of human-driven crossbreeding. Male ligers can reach 11.8 feet long and weigh over 1,100 pounds, bigger than either parent species. This unnatural size results from conflicting genetic growth regulation between lions and tigers. Male lions' genes promote maximum offspring growth, while female lions carry growth-limiting genes. Tigers lack these checks and balances. When combined, the lion's growth-promoting genes run unchecked in the tigress, creating gigantic offspring. This extreme size comes with severe health consequences. Ligers suffer from gigantism, causing organs to struggle, keeping pace with body growth, leading to shortened lifespans. Many develop neurological disorders, obesity, genetic defects, and die before reaching 7 years old. Birth presents dangers, with tigresses experiencing difficult deliveries requiring cesarean sections. All male liger are sterile, though some females can reproduce, creating secondary hybrids like ligers. Liger only exist in captivity, consuming 25 pounds of meat daily. A monstrous appetite for a creature born entirely from human manipulation. 11. Modern broiler chicken. The chicken on your dinner plate is drastically different from chickens 50 years ago. Warped by decades of selective breeding into something almost unearthly. Modern broiler chickens like the Ross 308 have been selectively bred to grow at terrifying speeds. These industrial birds reach slaughter weight in less than 6 weeks, gaining over 60 g per day. In 1957, chickens took 16 weeks to reach market weight. This represents one of the most dramatic transformations in animal breeding history, achieved entirely through selective breeding. But this acceleration comes at a horrific cost. A chicken like this will put on more than 100 g of weight a day. The bird's muscle grows so fast that bones, hearts, and lungs cannot keep up. Their hearts and lungs are proportionally smaller than slower-growing breeds, yet must support much larger bodies. This leads to sudden death syndrome, where birds collapse dead from heart failure, affecting up to 35% of flocks. The rapid muscle growth causes leg problems so severe that many birds can barely walk. Lameness affects a significant portion, leaving them sitting in waste, developing painful footpad dermatitis. Their breast muscles grow so quickly they develop wooden breast and white striping. We've engineered chickens so grotesquely large they can barely stand. Their bodies twisted by the relentless demands of human design. 10. Teacup pigs. The teacup pig rose to fame as a trendy pet, flaunted by celebrities as tiny, eternal piglets, an illusion that masks the dark reality of their engineered fragility. Breeders promised pigs remaining under 50 pounds, perfect apartmentist companions. The reality was far different and created an animal welfare crisis. Most teacup pigs aren't a distinct breed, but pot-bellied pigs sold as young piglets. Unethical breeders use several deceptive tactics. They show customers the smallest runt, claiming it represents adult size. They deliberately underfeed breeding pigs to stunt growth, showing emaciated parents as proof of small size. They sell piglets at just weeks old when any pig is tiny. Buyers discover the truth within months. Their micro pig continues growing, often reaching 150 to 200 pounds or more over 3 years. What seemed like a cute lap pet becomes a full-sized hog, requiring extensive space and specialized care. The pigs themselves suffer tremendously. Many experience malnutrition from deliberate underfeeding by owners desperate to keep them small. They develop painful joint problems, organ damage, and behavioral issues. Confined to apartments, they become destructive and aggressive. Thousands are abandoned, left to languish in overcrowded sanctuaries. A grim aftermath of humanity's relentless pursuit of unnatural perfection. 9. Savannah cat. Cross a domestic cat with an African serval and you unleash the Savannah cat. One of the largest and most unnerving domestic cat breeds, a predator cloaked in human design, these hybrids can reach 25 pounds with long legs, large ears, and striking spotted coats, maintaining their wild appearance. They were first bred in the 1980s, combining the exotic look of a wild cat with supposedly domestic temperament. Genetics, it's an organism that's made up of at least 2 different sets of DNA. Savannah cats exhibit behaviors making them challenging and sometimes dangerous pets. They possess incredibly strong prey drives, jumping abilities up to 8 feet, and intelligence letting them open doors and cabinets. First-generation Sauvas retain significant wild behaviors, including aggression, marking territory, and unpredictable temperaments. The breeding process itself raises ethical concerns. Servals and domestic cats are separated by millions of years of evolution. Mating often requires artificial insemination. Pregnancy complications are common with many kittens dying shortly after birth. Survivors may have health problems including hypertrophic cardiomyopathy, a silent creeping threat born from their unnatural hybrid origins. 8. Envir pig. At the University of G in Canada, scientists engineered the envir by splicing genes from E. coli bacteria and mice directly into pig embryos. A startling fusion of species that defies nature. This genetically modified pig produces an enzyme in its salivary glands that breaks down phosphorus in its feed more efficiently. The result was Enviro pigs producing up to 75% less phosphorus in their manure compared to conventional pigs. The environmental problem is real. Large-scale pig farming creates massive phosphorus pollution when manure runoff contaminates water systems, causing algal blooms that kill aquatic life. The Enviro pig seemed like an elegant solution using genetic engineering to address industrial agriculture's environmental damage. But the project raised troubling questions about solving problems with more problems. Rather than addressing fundamental issues with factory farming scale and waste management, scientists created GM animals to accommodate unsustainable practices. Safety concerns centered on unknown long-term health effects and potential ecosystem impacts if transgenic pigs escaped and bred with wild populations. The inserted bacterial and mouse genes created an organism nature never intended. The Enviral Pig program ultimately failed. After spending millions in development, Canadian regulators never approved the pigs. By 2012, funding dried up and researchers euthanized the entire research herd. Chilling end to creatures born from human ambition. 7. Anchor mouse. In 1988, Harvard University received the first patent ever granted for a genetically modified animal. The anchor mouse carries a human cancer gene that makes it highly susceptible to developing tumors. Scientists created this mouse specifically to suffer from cancer for research purposes. The genetic modification introduces an activated oncogene into the mouse's genome, ensuring it will develop cancer rapidly and predictably. This makes oncomus valuable for testing cancer treatments and studying tumor development. Pharmaceutical companies and research institutions worldwide use thousands of these mice annually. The ethical implications are staggering. We've deliberately created an animal whose purpose is to experience a terrible disease. Every anchor mouse is born with a guarantee of developing painful tumors. They exist solely to be afflicted, experimented upon, and killed. Defenders argue encomas have contributed to crucial cancer research leading to treatments saving human lives. Critics counter that creating animals designed to suffer represents humanity at its most troubling, turning sentient beings into disposable research tools through genetic manipulation. 6. Human pig chimera. In 2017, scientists at the Salk Institute created embryos that were part human, part pig. They injected human stem cells into pig embryos which developed human cells within pig bodies for several weeks before being destroyed. The dream of generating tissues and organ for transplantation into human. The goal was growing human organs inside pigs for transplantation, potentially solving the organ donation shortage. The chimera embryos developed normally with human cells integrating into pig tissue. Researchers terminated the experiments at 28 days, but embryos showed human cells beginning to specialize into different organ types. Scientists are working toward creating pigs whose organs could be harvested for human transplant patients. The ethical concerns are profound and existential. What happens if human brain cells develop in the pig's brain? Could this create a pig with partial human consciousness? At what point does a chimera deserve human rights or pig rights? Practical concerns compound. Ethical ones. Could diseases jump between species through these chimeras? What becomes of a pig carrying human organs throughout its body? Multiple countries ban human-animal chimera research. Others allow it under strict limits. These experiments tear through nature's limits, creating abominations that defy evolution and serve as a dark warning of how far humanity can play God. 5. Genetically modified mosquito. Aedes aegypti. They look like ordinary mosquitoes, but their DNA carries a secret designed by scientists. These are genetically modified Aedes aegypti mosquitoes created to stop the spread of deadly diseases like dengue, Zika, and yellow fever. Using advanced genetic engineering, researchers inserted a self-limiting gene that prevents offspring from surviving to adulthood. Only males, which don't bite humans, are released into the wild. When they mate with wild females, their young die before maturing, slowly collapsing mosquito populations over time. The results are both promising and controversial. Field trials in places like Brazil and Florida show steep drops in mosquito numbers. Yet, critics warn of unforeseen consequences. What if the gene spreads beyond control or disrupts ecosystems that depend on mosquitoes for food? The project represents a powerful, unsettling idea that we can fight disease by rewriting life itself. But can humanity survive the horrors it unleashes by rewriting the very fabric of life itself? 4. Dolly the sheep. In 1996, scientists at the Roslin Institute successfully cloned a mammal from an adult cell for the first time. Dolly became the world's first cloned animal, prompting a huge ethical debate. Dolly the sheep was genetically identical to her 6-year-old parent, created using somatic cell nuclear transfer. A cell nucleus from an adult mammary gland was inserted into an egg whose nucleus had been removed. That egg developed into Dolly. Dolly proved that adult specialized cells could be reprogrammed to create an entire organism. This achievement revolutionized biology and promised applications from preserving endangered species to growing replacement organs. But it also opened questions regarding cloning ethics. Dolly's creation required 277 attempts. Most cloned embryos died at various developmental stages. This high failure rate involving immense suffering for surrogate mothers and failed clones remains a serious ethical problem with cloning technology decades later. Dolly herself developed arthritis at a relatively young age and died at 6, about half the normal lifespan for her breed. Scientists debated whether her early aging resulted from being cloned from a 6-year-old cell. Other cloned animals exhibit the same eerie fate, withered and aging far too soon, as if nature itself rebels against human tampering. 3 gene-edited hornless dairy cows. Using CRISPR gene editing technology, scientists remove genes responsible for horn growth in dairy cattle, creating hornless cows without traditional breeding. This genetic modification eliminates the need for dehorning, a painful procedure where farmers burn or cut off calves' horn buds to prevent injuries. From an animal welfare perspective, this seems positive. Dehorning causes significant pain and stress. Engineering hornless cattle could prevent suffering for millions of animals. The gene-edited cows appear healthy and pass the hornless trait to offspring, solving the problem permanently. But the FDA discovered an unexpected complication. The edited cows also carried bacterial DNA accidentally inserted during the gene editing process. This contamination came from the DNA template used to make the edit, revealing that CRISPR isn't as precise as believed. The bacterial genes persisted in the cattle's genomes and were inherited by offspring. This discovery halted regulatory approval and revealed a concerning possibility. Genetic changes from editing could have unforeseen consequences. We could be unleashing animals with hidden twisted mutations, nightmares lying dormant, ready to surface in future generations when it may be too late to stop them. 2 designer dogs with genetic diseases. The obsession with perfect looks has twisted countless dog breeds into fragile shadows of themselves burdened with relentless genetic diseases. Cavalier King Charles Spaniels, bred for small skulls, have brains too large for their heads, causing syringing and excruciating headaches. German Shepherds suffer degenerative myelopathy from slope backs bred for show rings. Pugs experience breathing difficulties so severe that many require surgery. Dachshunds' elongated spines cause intervertebral disc disease, leaving many paralyzed. Dalmatians have a genetic predisposition to deafness. Golden retrievers face cancer at alarming rates, with 60 dying from the disease. These aren't random mutations. Breeders deliberately selected for traits they found attractive or believed matched breed standards even as those traits brought suffering. The closed stud books of purebred registries prevent introducing outside genetic diversity that could alleviate these problems. Modern DNA testing reveals how genetically impoverished many breeds are. Bulldogs, pugs, and King Charles Spaniels are trapped in a genetic dead end. Their diversity so eroded that restoring their health may be an impossible nightmare. 1 broad-breasted white turkey. The turkey on your Thanksgiving table is a grotesque shadow of its ancestors. A bloated, unnatural monstrosity born from decades of human manipulation. Wild turkeys weigh about 10 to 20 pounds. The broad-breasted white turkey, bred for commercial meat production, reaches 50 pounds or more in just 18 weeks. Their breast muscles are so enormous that these birds can barely walk or stand. This represents perhaps the most extreme example of selective breeding's physical impact. Modern turkeys literally cannot fly, struggle to walk, and suffer from constant leg pain under their own weight. Their hearts and lungs can barely support their massive bodies, causing frequent organ failure and sudden death. But the most disturbing aspect is reproduction. These turkeys have been bred so large-breasted that males physically cannot mount females to mate. The industry relies entirely on artificial insemination. Every single commercial turkey is produced through human intervention because natural reproduction is impossible. This means these animals cannot exist without us. We've engineered a creature so warped by selective breeding that it can no longer even reproduce. A living testament to humanity's ruthless interference. Do you think humans have gone too far with genetic engineering, or is it simply progress? Drop your thoughts in the comments. Thanks for watching, and we'll see you in the next.